Hello, everyone. Welcome to a session where I'm going to go through something I wrote last month called Towards a Formalized Theory for the Computational Foundations of Medicine. You can find this on the web. I'm going to say what I was talking about there. So let's start off. A theory of medicine, question mark. As it's practiced today, medicine is almost always about particulars. This has gone wrong, this is how to fix it. But might it also be possible to talk about medicine in a more general, more abstract way, perhaps to create a framework in which one can study its essential features without engaging with all of its details? So my goal is to take the first steps towards such a framework. And in a sense, my central result is that there are many broad phenomena in medicine that seem at their core to be fundamentally computational and to be captured by remarkably simple computational models that are readily amenable to study by computer experiment. So I should make it clear at the outset that I'm not trying to set up a specific model for any particular aspect or component of biological systems. Rather, my goal is to kind of zoom out and create what one can think of as a meta model for studying and formalizing the abstract foundations of medicine. What I'll be doing builds on things I've done recently on using the computational paradigm to study the foundations of biological evolution. And indeed, in constructing the idealized organisms, we'll be using the very same class of biological basic computational models. But now, instead of considering idealized genetic mutations and asking what types of idealized organisms they produce, we're going to be looking at specific evolved idealized organisms and seeing what effect perturbations have on them. Roughly, the idea is that an idealized organism operates in its normal, healthy way if there are no perturbations, but perturbations can derail its operation and introduce what we can think of as disease. With this setup, we can then think of the sort of fundamental problem of medicine as being the identification of additional perturbations that can treat the disease and put the organism at least approximately back on its normal healthy track. So, as we'll see, most perturbations lead to lots of detailed changes in our idealized organism, much as perturbations in biological organisms normally lead to vast numbers of effects, say at a molecular level. But as in medicine, we can imagine that all we can observe, and perhaps all we care about, are certain coarse-grained features or symptoms. And the fundamental problem of medicine is then to work out from these symptoms what treatment, if any, will end up being useful. By the way, when I say symptoms, I mean the whole cluster of signs and symptoms and tests and so on that one might in practice use, say, for diagnosis. It's worth emphasizing again that I'm not trying here to derive specific actionable medical conclusions. Rather, my goal is to build a conceptual framework in which, for example, it becomes conceivable for general phenomena in medicine that in the past have seemed at best vague and anecdotal to begin to be formalized and studied in a systematic way. At some level, what I'm trying to do is a bit like what Darwinism did for biological evolution. But in modern times, there's a critical new element, the computational paradigm, which not only introduces all sorts of new powerful theoretical concepts, but also leads us to the practical methodology of computer experimentation. And indeed, much of what I'll talk about here is based on the often surprising results of computer experiments that I've done recently that give us kind of raw material to build our intuition and structure our thinking about fundamental phenomena in medicine. So let's talk about a minimal metamodel. How can we make a metamodel of medicine? So we need an idealization of biological organisms and their behavior and development. We need an idealization of the concept of disease for such organisms, and we need an idealization of the concept of treatment. So for our idealization of biological organisms, we'll use a class of simple computational systems called cellular automata that I happen to have studied since the early 1980s. So let me show you an example of that. Hopefully that's an example. There we go. If I can only get that to go away, we're in good shape. Oh, come on. Oh, that's got to go away. There we go. Okay. So What's going on here is that we're progressively constructing the pattern on the left, representing the development and behavior of our organism by repeatedly applying cases of the rules on the right, representing the idealized genome and other biochemical, etc., rules of our organism. Roughly, we can think of the pattern on the left as corresponding to the life history of our organism, growing, developing, and eventually dying as it goes down the page. And even though there's a rather organic look to the pattern, remember that the system we've set up isn't intended to provide a model for any particular real-world biological system. Rather, the goal is just for it to capture enough of the foundations of biology that it can serve as a successful meta-model to let us explore our questions about the foundations of medicine. So, looking at our model in more detail, we see that it involves a grid of squares or cells, computational, not biological, each having one of, in this case, 4 possible colors, white and 3 others. So we start from a single red seed cell on the top row of the grid, then compute the colors of cells on subsequent steps, i.e. on subsequent rows down the page, by successively applying the rules on the right. The rules here are basically very simple, but we can see that when we run them, they lead to a fairly complicated pattern, which in this case happens to die out. In other words, all the cells become white after exactly 101 steps. So, what happens if we perturb the system? So, on the left here, we're showing the system as above without perturbation. But let me see, I think this is not the right picture. There we go. So on the left, we're showing the system without perturbation. But on the right, we're introducing a perturbation, indicated by the arrow, by changing the color of a particular cell on step 16, leading to a rather different, even if qualitatively similar pattern. So here are the results of other perturbations to our system. Some perturbations, like the one in the second panel here, quickly disappear. In essence, the system quickly heals itself. But in most cases, even single-cell perturbations, like the ones here, have a long-term effect. Sometimes they can increase the lifetime of the organism, sometimes they will decrease it. And sometimes, like in the very last case shown here, they'll lead to essentially unbounded kind of tumor-like growth. So in biological or medical terms, the perturbations we're introducing are minimal idealizations of things that can happen to an organism in the course of its life. Sometimes the perturbation will have little or no effect on the organism, or at least they won't really hurt it, and the organism will live out its natural life or even extend it a bit. But in other cases, a perturbation can somehow destabilize the organism, in effect making it develop a disease and often making it kind of die before its time. But now we can formulate what we can think of as the fundamental problem of medicine. Given that perturbations have had a deleterious effect on an organism, can we find subsequent perturbations to apply that will serve as a treatment to overcome the deleterious effect? So let's see, the first panel here shows a particular perturbation that makes our idealized organism die after 47 steps. The subsequent panels then show various treatments, in other words, additional perturbations, second arrows that we're indicating here, that serve to at least keep the organism alive. And the final panel on the right is what the organism would be like if it hadn't had any perturbation at all. So in these later panels, the kind of life history of the organism gets closer to the healthy, unperturbed form that we're showing on the far right. And if our criterion is restoring overall lifetime, we can reasonably say that the treatment has been successful. But it's notable that the detailed life history and perhaps quality of life of the organism will essentially never be the same as before. As we'll see in more detail later, it's almost inevitably the case that there will be at least some and often many long-term effects of the perturbation plus treatment, even if they're not considered deleterious. So now we've got an idealized model of sort of the problem of medicine. What can we say about solving it? Well, the main thing is that we can get a sense of why it's fundamentally hard. And beyond anything else, the central issue is a fundamentally computational one, the phenomenon of computational irreducibility. given any particular cellular automaton rule with any particular initial condition, one can always explicitly run the rule step by step from that initial condition, see what happens. But can one do better? Experience with mathematical science might make one imagine that as soon as one knows the underlying rule for a system, one should in principle immediately be able to kind of solve the equations and jump ahead to work out everything about what the system does without explicitly tracing through all the steps. But one of the central things I discovered in studying simple programs back in the 1980s is that it's common for such systems to show what I call computational irreducibility, which means that the only way to work out their detailed behavior is essentially just to run their rules step by step and see what happens. So what about biology? One might imagine that with its incremental optimization, biological evolution would produce systems that somehow avoid computational irreducibility and, like simple machinery, have obvious, easy-to-understand mechanisms by which they operate. But in fact, that's not what biological evolution typically seems to produce. And instead, as I recently argued, what it seems to do is basically just to pull together randomly found lumps of irreducible computation that happen to satisfy its fitness criterion. And the result is that biological systems are full of computational irreducibility and mostly aren't straightforwardly mechanically explainable. So the presence of computational irreducibility is presumably also why theoretical biology, based on mathematical models, has always been so challenging. But okay, given all this computational irreducibility, how is it that medicine is even possible? How is it that we can know enough about what a biological system will do to be able to determine what treatment to use on it? Well, computational irreducibility makes it hard, but it's a fundamental feature of computational irreducibility that within any computationally irreducible process, there must always be pockets of computational reducibility. And if we're trying to achieve only some fairly coarse objective, like maximizing overall lifetime, it's potentially possible to leverage some pocket of computational reducibility to do this. And indeed, pockets of computational reducibility within computational irreducibility are what make many things possible, including having understandable laws of physics, doing higher mathematics, and so on. The diversity and classification of disease. So with our simple idealization of disease as the effect of perturbations on the life history of our idealized organism, we can start asking questions like, what is the distribution of all possible diseases? So to begin exploring this, here are the patterns generated with a random sample of the 4,383 possible single-point mutations to the idealized organism we've been talking about so far. So what we're seeing here is lots of different arrows that kind of make perturbations at different places in that pattern. So clearly there's a lot of variation in these life histories, in effect a lot of different symptomologies. Well, if we average them all together, we lose the detail and we just get something kind of close to the original. But if we look at the distribution of lifetimes, we can see that while it's peaked at the original value, which was 101 steps, it nevertheless extends to both shorter and longer values. Well, in medicine, or at least Western medicine, it's been traditional to classify things that can go wrong in terms of discrete diseases. And we can imagine also doing this in our simple model. But it's already clear from the array of pictures that we just saw, that we saw here, that this isn't going to be a straightforward task. I mean, we've kind of got a different detailed pattern for every different perturbation. So how should we group them together? Well, much as in medicine, it depends on what we care about. In medicine, we might talk about signs and symptoms, which in our idealized model, we can basically identify with features of patterns. And so as an example, we might decide that the only features that matter are the ones associated with, for example, the boundary shape of our pattern. So what happens to these boundary shapes with different perturbations? Well, here are the most frequent shapes found together with their probabilities. So we might think of these as representing common diseases of our idealized organism. But what if we look at all possible diseases, at least all the ones produced by single cell perturbations? Using the boundary shape as our way to distinguish diseases, we can find that if we plot the frequency of disease against rank, we get roughly a power law distribution. And yeah, it's not clear why it's a power law. So what are the rare diseases, i.e. the ones with low frequency, the high rank here, or low rank, depending on how you look at it? What are they like? Well, their boundary shapes can be quite diverse. But okay, can we somehow quantify all these diseases? For example, as a kind of imitation medical test, we might look at how far to the left the boundary of each pattern goes. With single point perturbations, 84% of the time, it's the same as in the unperturbed case. But there's a distribution of other kind of less healthy results. Here I plotted that on a log scale. So that's showing the offset, the frequency of different offsets to the left. So extreme examples of this. Oops. Oh, that's strange. Huh. Well, I had some examples of extreme examples here, but they seem to have gone missing. So we could kind of diagnose any pattern that goes further to the left than the unperturbed one as a case of, say, leftiness syndrome. It's kind of amusing. I'm a left-handed person, so I'm stuck with leftiness all the time. Anyway, we might imagine that if we set up enough tests, we could begin to discriminate between many discrete diseases. But somehow this seems quite ad hoc. So can we perhaps be more systematic, say by using machine learning? Well, let's say we look at each whole pattern and then place it in an image feature space, say a two-dimensional one. So here's an example of what we get. And the details of this depend on the particulars of the machine learning method we've used. Here it's the default feature space plot method in Wolfram language, but it's a fairly robust result that visually different patterns end up separated, so that in effect the machine learning is successfully automating some kind of visual diagnosis. And there's at least a little evidence that the machine learning will identify separated clusters of patterns that we can reasonably identify as sort of truly distinct diseases, even as the more common situation is that between any two patterns, there are intermediate ones that aren't neatly classified as one disease or the other. So, somewhat in the style of the Human International Classification of Diseases, ICD, we can try arranging all our patterns in a hierarchy, though it's sort of basically inevitable that we'll always be able to subdivide further, and there'll never be a clear point at which we can say we've classified all the diseases. By the way, in addition to talking about possible diseases, we also need to discuss what counts as healthy. We could say that our organism is only healthy if its pattern is exactly what it would be without any perturbation, the natural state, so to speak. But what probably better captures everyday medical thinking is to say that our organism should be considered healthy if it doesn't have symptoms or features that we consider bad. And in particular, at least after the fact, we might be able to say that it must have been healthy if its lifetime turned out to be long. It's worth noting that even in our simple model, while there are many perturbations that reduce lifetime, there are also perturbations that increase lifetime. In the course of biological evolution, genetic mutations of the overall underlying rules for our idealized organism might have managed to achieve a certain longevity. But the point is that nothing says that longevity perturbations applied during the life of the organism can't get further. Well, in fact, these are some examples of where they do. Remember, our original organism shown here on the left has 101, lives 101 steps. But with the perturbations, the kind of interventions shown in the other panels here, we succeed in making the system live longer. Well, actually, in a feature that's not, at least yet, reflected in human medicine, there are, as we can see, perturbations that can make the lifetime very significantly longer. And for the particular idealized organism we're studying here, the most extreme examples obtained with single point mutations are these ones here. Okay, well, what happens? So, for example, there are ones here that are living 3.5 times the natural lifetime, so to speak, and so on. Okay, but what happens if we consider perturbations at multiple points? There are immediately vastly more possibilities. So here are some examples of the 10 million or so possible configurations of 2 perturbations. Here are some examples with 3 perturbations. And here are some examples if we try to apply 5 perturbations, though sometimes the organism is kind of already dead before we can apply later perturbations. So what happens to the overall distribution of lifetimes in these cases? Already with 2 perturbations, the distribution gets much broader, and with 3 or more, the peak at the original lifetime has all but disappeared, with a new peak appearing for organisms that in effect die almost immediately. So, in other words, the particular idealized organism that we're studying is fairly robust against 1 perturbation, and perhaps even 2. But with more perturbations, it's increasingly likely to succumb to what we can think of as infant mortality. And yes, if one increases the number of perturbations, the life expectancy progressively decreases. What about the other way around? With multiple perturbations, can the organism in effect live forever? So here are some examples where it's kind of still going strong after 300 steps. And but after 500 steps, most of these have died out. Let's see what am I saying here. Okay. So we can see here most of these have died out, but as is pretty typical in the computational universe, perhaps like in medicine, there are always surprises, courtesy of computational irreducibility, like the sudden appearance of the obviously periodic case, the third one here, with period 25. And that's how it starts on the left, and that's how it ends up going on the right with period 25. As well as much more complicated cases where, let's see, we're looking at these here. These are cases where we've succeeded in we've made something which has become periodic. In the last case here, we've kind of rectified the picture to make it more vertical, to make it easier to see what's going on. But these are cases where adding these perturbations has caused the system to do all sorts of strange things. Not perhaps very medically relevant things, at least for humans, where the system has just become sort of infinitely periodic. But so, in other words, in these cases, the organism does in effect live forever, though not in an interesting way. Indeed, such cases might remind us of kind of tumor-like behavior in biological organisms. But what about a case that not only lives forever, but also grows forever? Well, needless to say, looking out in the computational universe, we can find an example. Here it is. So the incidence of this behavior, of the growing forever behavior, is about 1 in a million for 2 perturbations, or more precisely 7 out of 9.6 million possibilities, and 1 in 300,000 for 3 perturbations. And although there are presumably more complicated behaviors out there to find, they don't show up with 2 perturbations, and their incidence with 3 perturbations is below about 1 in 100 million. Well, let's talk about diagnosis and prognosis. So a fundamental objective in medicine is to predict from tests we do or symptoms and signs we observe what will happen. And yes, we now know that computational irreducibility inevitably makes this in general hard, but we also know from experience that a certain amount of prediction is possible, which we can now interpret as successfully managing to tap into pockets of computational reducibility. So as an example, let's ask what the prognosis is for our idealized organism based on the width of its pattern that we measure at a certain step. So here, for example, is what happens to the original lifetime distribution, which is shown in green here, if we consider only cases where the width of the measured pattern after 25 steps is less than its unperturbed healthy value. We're dropping the 1% of cases where the organism was already dead before 25 steps. So our sort of narrowed cases represent about 5% of the total. Their median lifetime is 57, as compared to the overall median lifetime of 106. But clearly the median alone doesn't tell the whole story, and nor do actually the 2 survival curves. So remember, this is a survival curve for our organism with a perturbation, that's the one in yellow, and this is our survival curve for instances of our organism that had a width below which were narrower after 25 steps than they were in the unperturbed case. So it's kind of the the natural course of all possible, in a sense, diseases is shown in yellow. The ones where we measured a symptom that the thing will be narrower than 25 steps is shown in green. So the ones which were narrower than 25 steps, the slimmer organisms after 25 steps lived longer. Well, so we can look at this survival curve doesn't really tell the whole story. So we can look at the actual widths as a function of time for all the narrow cases compared to the sequence of widths for the unperturbed case. And it's so the unperturbed case is the blue curve, and all the different possible perturbed cases are the orange ones. And we can see what the width is, and we can see what happens if we force it to be a certain width. So these pictures don't make it look promising that one could predict lifetime from the single test of whether the pattern was narrow at step 25. Well, like in analogous medical situations, one needs more data. One approach in our case is to look at the actual narrow patterns up to step 25. So here we've got a picture of that sorted by ultimate lifetime. And then so we're looking at the actual, let's see, we're looking at this. Oh dear, I seem to have a missing picture again. That's a shame. Let me see if I can't find that picture. Hold on 1 second. No, it is a profoundly missing picture. Oh well, humph. I don't know quite how that happened. Let me see here. Oh well. In any case, what I was trying to do here was to show you a picture of all the patterns sorted by width, and then to ask the question, how do we predict from the actual pattern up to size up to step 25 what will happen? So instead of just knowing the width, we know the whole pattern. So another thing we can do, in addition to looking at the whole pattern, kind of using machine learning and so on, it's of course difficult medically to sort of get the whole pattern. We tend to get just discrete measurements, at least in the current state of medicine. But a simple approach is not just to do a sort of a discrete narrow or not test, but rather to look at sort of the actual width at step 25. So, oops, that's not the right thing at all. Let me see if I can get to the right place. Here we go. So this is looking at the step at width 25 versus lifetime. So that's how much can we predict by knowing the step a width at step 25? How much can we predict of the lifetime? And so here's what we get if we look at the distribution of outcomes together with the median in each case. So this is the distribution of outcomes given the width at step 25. What will the lifetime be? These are the distributions. That's the median result. So the predictive power of our width measurement is obviously quite weak, although there's doubtless a way to kind of hack p-values to get at least something out. It would probably be, if I'm a little bit cynical, I would say that the abstract of the medical paper would still look fine, even though, to my eye, this looks like a very weak predictive power. Unsurprisingly, machine learning doesn't really help. Like here's, I hope, a machine learning prediction based on decision tree methods for lifetime as a function of width. And yes, it's very close to just saying the prediction is the median. Does it help if we use more history? In other words, what happens if we make our prediction not just based on the width at a particular step, but from the history of all widths up to that point? So As one approach, we can make a collection of training examples of what lifetimes particular width histories, say up to step 25, lead to. Here are some examples. And there's already something of an issue here because a given width history, which in a sense is a coarse graining of the detailed microscopic history, can lead to multiple different outcomes, as in these cases. But we can still go ahead and try to use machine learning to predict lifetimes from width histories based on training on, say, half of our training data. Well, we get sort of less than impressive results. If this was a perfect prediction, this would be kind of a straight line of actual versus prediction. So the vertical line here is associated with multiple lifetimes from a single width history in the training data. So how can we do better? Well, given the underlying setup for our system, if we could determine not just the width, but the whole precise sequence of values for all cells, even at step 25, then in principle, we could use this as an initial condition and run the system forward to see what it does. But regardless of it being medically implausible to do this, it isn't much of a prediction anyway. It's more just watch and see what happens. And the point is that insofar as there's computational irreducibility, one can't expect, at least in full generality, to do much better. And as I'll talk about later, there's no reason to think that organisms produced by biological evolution will avoid computational irreducibility at this level. But still, within any computationally irreducible system, as I said, there are always pockets of computational reducibility. So we can expect that there will be some predictions that can be made. But the question is whether those predictions will be about things we care about, like lifetime, or even about things we can measure. Or in other words, will they be predictions that speak to things like symptoms? Our physics project, for example, involves all sorts of underlying processes that are computationally irreducible. But the point there is that what physical observers like us perceive aggregate constructs, like overall features of space, that show significant computational reducibility. In a sense, there's an analogous issue here. There's computational irreducibility underneath, but what do medical observers actually perceive? And are there computationally reducible features related to that? If we could find such things, then in a sense we'd have identified general laws of medicine, much like we now have general laws of physics. The problem of finding treatments. So we've talked a bit about giving a prognosis for what will happen to an idealized organism that suffered a perturbation. But what about trying to fix it? What about trying to intervene with another treatment perturbation that can heal the system and give it a life history that's at least close to what it would have had without the original perturbation? Well, here's our original idealized organism, together with how it behaves when it suffers a particular perturbation that significantly reduces its lifetime. But what happens if we now try applying a second perturbation? Here are a few random examples. None of these kind of convincingly heal the system, but let's, as we can in our idealized model, just enumerate all possible second perturbations. Here there are 1,554 of them. Then it turns out that at least a few of these do in fact successfully give us patterns that at least exactly reproduce the original lifetime. And oh no. Oh, I'm showing you. Hold on. Dear, oh dear, oh dear. So that's our single perturbation. What I was trying to show you before were some random perturbations, but what I actually ended up with here are specially chosen perturbations out of the 1,554 possibilities. These are particular perturbations that are treatments that successfully reproduce the original lifetime. So do these represent true examples of healing? Well, it depends what we mean. Yes, they've managed to make the lifetime exactly what it would have been without the original disease-inducing perturbation, but in essentially all cases, we see here that there are various long-term side effects, in the sense that the detailed patterns generated end up having obvious differences from the original unperturbed healthy form. So there's The one exception here is the very first case on the left, in which, in a sense, the disease was caught early enough that the treatment perturbation manages to completely heal the effects of the disease perturbation. And we can kind of see that in action here. We see in more detail the bits being changed, and on the right, we're showing kind of the difference of the total difference, which after a few steps goes to zero. So we've been talking here about intervening with treatment perturbations to heal our disease perturbation. But actually, it turns out there are plenty of disease perturbations which automatically heal themselves without any treatment intervention. In fact, of all possible 4,383 single perturbations, 380 essentially heal themselves. In many cases, the healing happens very locally after one or two steps. We're seeing here where there are changes, and we see that the changes die out quickly. But there are also more complicated cases where perturbations produce fairly large-scale changes in the patterns. We can see that down there, that nevertheless spontaneously heal themselves. We can see the changes down in the panels below, where they're kind of blacked out squares. Well, needless to say, in cases where a perturbation spontaneously heals itself, adding a treatment perturbation will almost always lead to a worse outcome. So how should we think about perturbations that spontaneously heal themselves? They're like seeds for diseases that never take hold, or like diseases that quickly burn themselves out. But from a theoretical point of view, we can think of them as being where the unperturbed life history of our idealized organism is acting as an attractor to which certain perturbed states inexorably converge, a bit like how friction can dissipate perturbations to patterns of motion in a mechanical system. But let's say we have a perturbation that doesn't spontaneously heal itself. Then to remediate it, we have to do the medical thing, and in our idealized model, try to find a treatment perturbation. So how might we systematically set about doing that? Well, in general, computational irreducibility makes it difficult. And as one indication of this, this shows, let's see, what lifetime can be achieved by treatment perturbations made at each possible point in the pattern after the initial perturbation. So what we're showing here on that right-hand panel, the color is showing if we make a perturbation at that point, what lifetime can we achieve? So red means actually that we have sort of a tumor-like behavior of the thing growing forever. We see that sort of darker orange means we managed to live a long time, at least as long as the pattern on the left. So we can think of this as providing a map of what the effects of different treatment perturbations will be. So here are a few other examples from different initial perturbations on, in effect, different diseases. And we see that quite a lot, quite often here, we're getting kind of sort of tumor-like behavior of the dark red things where the pattern lives forever. And sometimes we're getting behavior where the pattern will successfully sort of live for as long as we want it to. So there's some regularity here, but the main observation is that different detailed choices of treatment perturbations will often have very different effects. In other words, even nearby treatments will often lead to very different outcomes. Given computational irreducibility, this isn't surprising, but in a sense, it underscores the difficulty of finding and applying treatments. By the way, cells indicated in this, as I said, in the dark red are ones where treatment leads to a pattern that lives excessively long, or in effect, as I was saying, leads to sort of tumor-like characteristics. And the fact that these are scattered so seemingly randomly reflects the difficulty of predicting whether such effects will occur as a result of treatment. In what we've done so far, our treatment has always consisted of just a single additional perturbation. But what about applying more perturbations? For example, let's say that we do a series of experiments where our first treatment perturbation, after our first treatment perturbation, we progressively try other treatment perturbations. So if a given initial perturbation doesn't get further from the desired lifetime, we keep it. Otherwise, we reject it and try another perturbation. So this is an example of what happens if we do this. So the panels that highlighted here represent perturbations that we kept. And there's a typo here. Here's how the overall lifetime kind of converges in successive iterations of our experiment. So it's kind of like a fitness curve or a loss curve that we're seeing here. So in what we just did, we allowed additional treatment perturbations to be added at any subsequent step. But what if we require treatment perturbations to always be added on successive steps, starting right after the disease perturbation occurred? So here's an example of what happens in that case. And here's what we see if we kind of zoom in. Oh, no. Yet again, I'm sorry, we have something missing here. Let me see. I wonder if I do something here, whether I can get this to find my other slides. Let me try something here. Let me do this. I'm going to kill that. I'm going to try and see if I can debug this a little bit. That's not working. Oh, wow. That's really weird. I need to get these synced onto this machine. Those should be synced. Okay. Let me try. That doesn't seem to make a lot of sense there, but let me try this. Wait a minute, what did that do? Hold on a second, everybody. Let's see if I can debug this. I am going to quit this entirely. Oh, wait a minute, maybe this worked. Okay. Oh, that didn't work because it's going to the wrong place. Let me see whether I can set this correctly. Oh. Hold on. There we go. Okay. That was the picture I was looking for. So, this is disease perturbations added. This is treatment perturbations added right after the disease perturbation occurred. Okay, so now, let's see. That's not right there. Let's try and find what I'm doing here. Oh, look, that was a slide I couldn't show you before, showing as a function of showing different possible configurations that have a certain width. Okay, let's go back to where I was. Okay, so it's kind of zooming in on the early steps where we're giving perturbations at successive steps after we're giving treatment perturbations at successive steps after the disease perturbation. In a sense, this corresponds to sort of doing aggressive treatment as soon as the initial disease perturbation has occurred. A notable feature of this particular example is that when our succession of treatment perturbations have succeeded in restoring the lifetime, which actually happens fairly quickly, the life history they produce is similar, though not identical, to the unperturbed case. And we can kind of see that here. So that definitely doesn't always happen. So, for example, here are cases where that hasn't happened, where we have recovered something close to the lifetime, but the actual form of the organism is very different. So it's worth pointing out that if we allowed ourselves to do many single perturbations at the same time, i.e. on the same row of the pattern, we could effectively just define new initial conditions for the pattern and, for example, perfectly regenerate the original unperturbed pattern after this reset. And in general, we can imagine that in effect, hot wiring the organism by applying large numbers of treatment perturbations that just repeatedly direct it back to its unperturbed form. But such extensive and detailed intervention that in effect replaces the whole state of the organism seems far from what might be practical in typical current medicine, except perhaps in some kind of regenerative treatment approach. And indeed, in actual current medicine, one is normally operating in a situation where one doesn't have anything close to perfect cell-by-cell information on the state of the organism, and instead one has to figure things out like what treatment to give based on much coarser symptom-level information. In some ways, though, the immune system does something closer to cell-by-cell treatment. So what can one do given coarse-grained information? As one example, let's consider trying to predict what treatment perturbation will be best using the kind of pattern width information we were talking about before. Specifically, let's say we have the history of the overall width of a pattern up to a particular point. Then from this, we want to predict what treatment perturbation will lead to the best lifetime outcome for the system. There are a variety of ways we could approach this, but one is to make predictions of where to apply a treatment perturbation using machine learning trained on examples of optimal such perturbations. It's analogous to what we did before in applying machine learning to predict lifetime from width history, but now we want to predict from width history what treatment perturbation to apply. So to generate our training data, we can search for treatment perturbations that lead to the unperturbed lifetime when starting from life histories with a given width history. Now we can use simple neural nets to create a predictor that tries to tell us from a width history what treatment to give. So here are some comparisons between our earlier kind of search results based on looking at complete life histories and shown with the red arrows here. So what we're showing here with red arrows are the machine learning predictions based purely on width history before the original disease perturbation. So it's clear the machine learning is doing something, though it's not as impressive as perhaps it looks because the wide range of perturbations all in fact give rather similar life histories. So as a slightly more quantitative indication of what's going on, here's the distribution of lifetimes achieved by our machine learning-based therapy. Our best treatment was able to give lifetime 101 in all these cases here, and while the distribution we've now achieved looks sort of peaked around the unperturbed value, yeah, so this is the best treatment gave lifetime 101, but the distribution we've actually achieved here looks peaked around the unperturbed value. Dividing this distribution by what we get without any treatment at all makes it clear that not so much was achieved by the machine learning we were able to do. In a sense, this isn't surprising. Our machine learning, based as it is on coarse-grained features, is quite weak compared to the computational irreducibility of the underlying processes at work. Let's talk about the effect of genetic diversity. So what we've talked about so far, we've studied just a single idealized organism with a single set of underlying genetic rules. But in analogy to the situation with humans, we can imagine a whole population of genetically slightly different idealized organisms with different responses to perturbations and so on. Many changes to the underlying rules for our idealized organism will lead to unrecognizably different patterns that don't, for example, have the kind of finite but long lifetimes we've been interested in. But it turns out that in the rules for our particular idealized organism, there are some specific changes that actually don't have any effect at all, at least on the unperturbed pattern of behavior. And the reason for this is that in generating the unperturbed pattern, these particular cases in the rule just never happen to be used. So those are the cases that don't get used. So the result is that any one of the 4 cubed or 64 possible choices of outcomes for these cases that I've highlighted here will still yield the same unperturbed pattern. If there's a perturbation, however, different cases in the rule can be sampled, including these ones. It's as if cases in the rule that are initially non-coding end up being coding when the path of behavior is changed by a perturbation. Or said differently, it's like different genes being activated when conditions are different. So to make an idealized model of something like a population with genetic diversity, we can look at what happens with different choices of our sort of initially non-coding rule outcomes. So here are some cases where we've made different choices of the non-coding parts of the rule, the unperturbedly non-coding parts of the rule. So before the perturbation happens, all these inevitably show the same behavior. So that's above where the arrow shows up. All these are showing the same behavior because they're never sampling non-coding rule cases. But as soon as there's a perturbation, the pattern is changed. And after varying numbers of steps, previously non-coding rule cases do get sampled and can affect the outcome. So here are the distinct cases of what happens in all 64 genetic variants, in which the red arrow in each case indicates where the pattern first differs from what it is with our original idealized organism. So in each of these cases, there are and we're underneath the numbers underneath say the multiplicity of examples out of all those 64 possible rules where this is the picture you get. But this is showing sort of where the change in the underlying rule starts having an effect on the behavior of the organism. So this is now the distribution of the lifetimes achieved, in effect showing sort of the differing consequences of this particular disease perturbation on all our genetic variants. So what happens with other disease perturbations? So here's a sample of distributions of lifetimes achieved where kind of just the flat line corresponds to cases where all 64 genetic variants yield the same lifetime. So this is showing for a sample of genetic variants how the lifetime differs with different perturbations. So what about the overall lifetime distribution across all single perturbations for each of the genetic variants? The detailed distribution we get, as we can see here, is different for each variant, but their general shape is always remarkably similar. Though taking differences from our original idealized organism reveals some structure. So so this is showing us how, for our different genetic variants, what are the lifetime distributions under certain perturbations for our organisms. It's showing if we had a population, it's showing us in what the genetic diversity of that population, what effect that has on the outcomes of certain, quotes, diseases or perturbations. So as another indication of the effect of genetic diversity, we can plot the survival curve averaged over all perturbations and compare the case for our original idealized organism with what happens if we average equally over all 64 genetic variants. So the difference is small, but there's a longer tail for the average of genetic variants than for our specific original idealized organism. So in other words, the average of the population lives slightly longer than our specific original particular organism. So we've seen a bit how our idealized genetic variation affects disease, but how does it affect treatment? For the disease we were looking at before, we already saw that there's a particular treatment perturbation that successfully returns our original idealized organism to its natural lifespan. So what happens if we apply the same treatment across all genetic variants? In effect, this is like doing a very idealized clinical trial of our potential treatment. And what we see is that the results are quite diverse, indeed more diverse than from the disease on its own. So in essence, what we're seeing is that, yes, there are some genetic variants for which the treatment still works, but there are many for which there are often fairly dramatic side effects. Biological evolution and our model organism. So where did the particular rule for our model organism we've been studying come from? Well, we evolved it using a slight generalization of the idealized model for biological evolution that I recently started talking about. The goal of our evolutionary process was to find a rule that generates a pattern that lives as long as possible, but not infinitely long, and that does so robustly, even in the presence of perturbations. In essence, we used lifetime, or more accurately, lifetime under perturbation, as our fitness function, then progressively evolved our rule, or genome, by random mutations to try to maximize this fitness function. In more detail, we started from the kind of null, everything turns white rule, then successively made random changes to single cases in the rule, point mutations, keeping the resulting rule whenever the pattern it generated had a lifetime under perturbation that wasn't smaller or infinite. So with this setup, this is the particular random sequence of rules we got, showing in each case the outcome for each of the 64 cases in the rule. Well, many of these rules sort of don't make progress in the sense that they increase the lifetime under perturbation, but every so often there's a breakthrough and a rule with a longer lifetime under perturbation is reached. And this is the sequence of kind of breakthroughs along underneath them showing the value of their lifetime. So as we can see, the rule for the particular model organism we've been using here is what's reached at the end. But in studying my recent idealized model for biological evolution, I considered fitness functions like lifetime that can directly be computed just by running the underlying rule from a certain initial condition. But here I'm generalizing that a bit and considering as a fitness function not just lifetime, but lifetime under perturbation, computed by taking a particular rule and finding the minimum lifetime of all patterns produced by it with a certain random perturbations applied. So for example, here the lifetime under perturbation in this picture here would be considered as the minimum of the lifetimes generated with no perturbation and with certain random perturbations, or in this case, the value that fitness value would be 60. So then, okay, this shows how the lifetime under perturbation fitness, indicated by the blue line, behaves in the course of our adaptive evolution, right around where the fitness 60 breakthrough occurs. So remember, we had back here on the second row, we have a fitness 60 breakthrough. It goes from 49 to 60. And so here we're showing what the actual series of perturbations that we tried were, the actual series of mutations in the genome that we tried to achieve that perturbation. And what's happening here is at each adaptive step, we're considering a new rule obtained by a point mutation from the previous one. And running this rule, we get a certain lifetime. If this lifetime is finite, we indicate it by a green dot. Then we apply a certain set of random perturbations, indicating the lifetimes we got from those by gray dots. We could imagine using all sorts of schemes for picking the random perturbations. Here what we're doing is just to perturb random points on about 1/10 of the rows in the unperturbed pattern. Okay, then the minimum lifetime for any given rule we're indicating here by a red dot. And this is the fitness that we assign to that rule. So now we can see sort of the whole progression of our adaptive evolution process. And one thing that's notable is that the unperturbed lifetimes, green dots, are considerably larger than the final lifetimes, red dots. And what this means is that our requirement of robustness, implemented by looking at lifetime under perturbation rather than just unperturbed lifetime, considerably reduces the lifetimes we can reach. In other words, if our idealized organism is going to be robust, it won't tend to be able to have as long a lifetime as it could if it didn't have to worry about random perturbations. So to illustrate this, here's a typical example of a much longer lifetime obtained by adaptive evolution with the same kind of rule we've been using, 4 colors, range 1, cellular automaton, but now with no perturbations and with fitness being given purely by the unperturbed lifetime, exactly as in what I did recently on biological evolution. Okay, so given that we're evolving with a lifetime under perturbation fitness function, what are some alternatives to our particular model organism? So here are a few examples. At an overall level, these seem to react to perturbations much like our original model organism. So here are some examples of that. One notable feature here, though, is that there seems to be a tendency for simpler overall behavior to be less disrupted by perturbations. In other words, idealized diseases seem to have less dramatic effects on simpler idealized organisms. And we can see a reflection of this phenomenon if we plot the overall kind of single perturbation lifetime distributions for these particular 4 rules that I'm showing here. So these are those lifetime distributions. Let's remember the rules here, the kind of the one at the bottom and the second one, those are sort of slightly simpler in the form of patterns that they're producing. And the third one, for example, is more complicated. So there are detailed differences here, but the main conclusion seems to be that there's nothing special about the particular model organism that we've used. And if we repeated our whole analysis for different model organisms, in other words, different idealized species, the results we'd get would be very much the same. Well, so what does this mean, and where do we go from here? Well, at the outset, it wasn't clear that there'd be a way to usefully capture anything about the foundations of medicine in a formalized theoretical way. But in fact, what we found is that even the very simple computational model we've studied seems to successfully reflect all sorts of features of what we see in medicine. Many of the fundamental effects and phenomena are, it seems, not the result of the details of biomedicine, but instead are at their core purely abstract and computational, and therefore accessible to formalized theory and metamodeling. So this kind of methodology is very different from what's been traditional medicine and isn't likely to lead directly to specific practical medicine. But what it can do is to help us develop powerful new general intuition and ways of reasoning, and ultimately an understanding of the conceptual foundations of what's going on. At the heart of much of what we've seen is the very fundamental and ubiquitous phenomenon of computational irreducibility. I've argued recently that computational irreducibility is central to what makes biological evolution work, and that's inevitably imprinted on the core computational architecture of biological organisms. And it's this computational irreducibility that inexorably leads to much of the complexity we see so ubiquitously in medicine. Can we expect to find a simple narrative explanation for the consequences of some perturbation to an organism? In general, no, because of computational irreducibility. There are always pockets of computational reducibility, but in general, we can have no expectation that, for example, we'll be able to describe the effects of different perturbations by neatly classifying them into certain set of distinct diseases. To a large extent, the core mission of medicine is about treating diseases, or in our terms, about remediating or reversing the effects of perturbations. And once again, computational irreducibility implies that there's inevitably a certain fundamental difficulty in doing this. It's a bit like with the second law of thermodynamics, where there's enough computational irreducibility in microscopic molecular dynamics that to seriously reverse or outpredict this dynamics is something that's at least far out of range for computationally bounded observers like us. In our medical setting, the analog is that computationally bounded interventions can only systematically lead to medical successes insofar as they tap into pockets of computational reducibility. And insofar as they are exposed to overall computational irreducibility, they will inevitably seem to show a certain amount of apparent randomness in their outcomes. In traditional approaches to medicine, one ultimately tends to give in to the randomness and go no further than to assign probabilities to things. But an important feature of what we've done here is that in our idealized computational models, we can always explicitly see what's happening inside. Often, largely as a consequence of computational irreducibility, it's complicated. But the fact that we can see it gives us the opportunity to get much more clarity about the fundamental mechanisms involved. And if we end up summarizing what happens by giving probabilities and doing statistics, it's because this is something we're choosing to do, not something we're forced to do because of our lack of knowledge of the systems we're studying. Well, there's much more to do in our effort to explore the computational foundations of medicine, but already there are some implications that are beginning to emerge. Much of the workflow of medicine today is based on classifying things that can go wrong into discrete diseases. But what we've seen here, which is hardly surprising given practical experience with medicine, is that when one looks at the details, a huge diversity of things can happen whose characteristics and outcomes can't really be binned into neatly discrete diseases. Well, indeed, when we try to figure out treatments, the details matter. As a first approximation, we might base our treatments on coarse graining into discrete diseases, but as what I've sort of outlined here can potentially help us analyze, the more we can directly go from detailed measurements to detailed treatments through computation, machine learning, and so on, the more promising it's likely to be. Not that that's easy, because in a sense, we're trying to beat computational irreducibility with computationally bounded measurements and interventions. In principle, one can imagine a future in which our efforts at treatment have much more computational sophistication, and indeed the immune system presumably already provides an example in nature. We can imagine things like algorithmic drugs and artificial cells that are capable of amounts of computation that are a closer match for the irreducible computation of an organism. And indeed, the kind of formalized theory that I've been talking about here is likely what one needs to begin to get an idea of how such an approach might work. In the thermodynamic analogy, what we need to do is a bit like reversing entropy increase by sending in large numbers of smart molecules. By the way, seeing how difficult it is potentially to reverse the effects of a perturbation provides all the more impetus to consider starting from scratch, as nature does in successive generations of organisms, and simply wholesale regenerating elements of organisms rather than trying to fix what's there. And yes, in our models, this is, for example, like starting to grow again from a new seed and letting the resulting pattern knit itself into the existing one. One of the important features of operating at the level of computational foundations is that we can expect the conclusions we can draw to be very general. We might wonder whether perhaps the framework we've described here could be applied outside of medicine. And to some extent, I suspect it can, potentially to areas like robustness of large-scale technological and social systems, and specifically things like computer security and computer system failures. And yes, much as in medicine, one can imagine, for example, classifying diseases for computer systems. But things won't likely be quite the same in these cases because the underlying systems have much more human-determined mechanisms and much less blind adaptive evolution. But when it comes to medicine, the very presence of computational irreducibility introduced by biological evolution is what potentially allows one to develop a robust framework in which one can draw conclusions purely on the basis of abstract computational phenomena. So what I've done here just begins to scratch the surface of what's possible. But I think we can already see that seen enough that we can be confident that medicine is yet another field whose foundations can be seen as fundamentally rooted in the computational paradigm. Thanks. That's what I wrote. So let's see. I'm happy to try and address maybe a few questions and comments. Let's see. Let's see. P. Bezakov asks, how can you explain the success of smallpox vaccines? In that case, human computation has won from nature, the immune system. Well, gosh golly, the immune system is a somewhat different story from what I've been talking about here, because the immune system itself is an example of molecular scale computation. The question of what happens when you introduce an antigen, you get responses, you select different collections of cells and antibodies and so on from the immune system, that's kind of itself an example of computational, of molecular scale computation. And what I've been more addressing here is things where you're not getting inside and doing sort of large scale molecular computation comparable to the level of computation that's sort of happening in the natural course of development of the organism. So I think it's really sort of a separate question. There's all kinds of computational analysis that one can do for the immune system, but that's really a different story. Let's see. Matthew asks, what is the criteria for the foundation of medicine to begin with? Can we uniformly describe health and illness accurately enough to build any kind of model that isn't completely general? Well, I think that's one of the things that comes out of what I've been studying here. What do we mean by being healthy? You know, looked at from the outside, we may live a completely satisfactory life, even though, and everything may be great, and we maybe feel very healthy, even though there's something about some random organ of ours that's weird with respect to the way that sort of the average human behaves. It may not matter to the things we care about, but it could be that, you know, there's something, I don't know, there's some, you know, feature of some muscle or tendon or some such other thing that in the normal course of a simple tech CEO's life, for example, doesn't make any difference. But if one was sort of doing some, you know, competitive long jumping or something, that would cause one trouble. So it's kind of this question of whether whether we consider ourselves healthy depends a bit on what we're trying to do with what we have, so to speak. And I think it's also interesting to see, and it's sort of a trap in a sense in practical medicine sometimes, that you know, you cure the disease but kill the patient type thing. You nominally solve some problem, like you nominally increase the lifetime, but the organism, the actual life history of the organism is terribly different than it would have been before in a way that the patient, for example, may consider quite unsatisfactory. So it is a non-trivial thing to define what we mean by healthy. And I talked about that a bit of we have these criteria there. We realize from these observations, from this kind of exploration, that we do have to know what we mean by healthy. You know, we're not going to get precisely the same pattern of bits, so to speak. It's going to be a bit different, but it may have features that we consider to be satisfactory features, like we live a long time, we have certain, you know, we're looking at the pattern and it has a certain width and so on, which is sort of a proxy for some kind of feature of what the organism can do. But yes, that's a really good question, and something that I think is really, you know, we're kind of shining a bright light on that question with these kinds of explorations. I'm not quite sure I understand this question from Drew. Will there be different formulations for each scientific perspective? I'm not quite sure what that means. So there's a comment from Prometheus saying some diseases are kind of categorical, like type 1 diabetes, and some more continuous, like type 2 diabetes. And these might seem to require different approaches. So, you know, say perturbations are interesting. I wonder how many diseases are due to perturbations and others no perturbations, but the pattern just dies off for reasons forever beyond our computation. Well, I mean, talking about, let's say, type 1 diabetes and type 2 diabetes, I mean, type 1 diabetes, the immune system kills off the beta cells. You don't produce insulin. You need to get it from the outside. Type 2 diabetes, you're kind of becoming insulin resistant, and you need more insulin to be able to sort of operate in the same way. There are, I think, sort of combinations, type 2 diabetes, type 1 diabetes that has sort of type 2 connected to it and so on. It is, like so many things in medicine, not black and white. I mean, I think one principle about medicine is very different from physical science, for example. In physical science, the simple explanation is usually the right one. In medicine, there's always another effect. There's always some complicated thing going on. There's always some special case. There's always some unexpected side effect and so on. And I think that's actually a reflection of the computational irreducibility that's kind of burnt into organisms like us from the way that biological evolution has managed to build us, so to speak. But, this question of whether if you have sort of the organism without perturbations, is that organism going to sort of live perfectly? I think that when I talk about perturbations, I'm kind of bundling together a whole bunch of different things. It could be what you eat. It could be something to do with what you were exposed to when, what happened when some particular cell division happened, in utero or some such other thing. I'm not really distinguishing sort of where the perturbation came from, what kind of a thing was happening. What I am saying is that there is, in a first approximation, sort of a natural course of the development of the organism. Now, for something like a C. elegans, a nematode, we know there are a couple of thousand cells and they just form in a particular pattern. At least the cells form in that pattern. The molecular structure of those cells will be different between every nematode, but at least at that level, the cells form in a definite pattern. For us humans, that's not the case. We have general organs and so on, but the details of, which particular brain cell is connected to which other one, those are not determined by the pure genetics of what's going on. Those are the result of things that happen to happen, so to speak, as the organism develops. So I'm just bundling all those things that happen to happen into this notion of a perturbation. It's a terrible approximation, but it gives us some idea of kind of what the sort of changes to things are like. We could as well just say, well, occasionally the rule is a bit different. That's pretty much exactly what we're saying. Occasionally, the rule isn't kind of what we thought the rule would be. Occasionally, that cell division works in a slightly different way, or some such other thing. It's a very kind of rough approximation. Now, when it comes to what originates different diseases, I mean, we don't know in most cases. I mean, we have some idea, but in the end, presumably something starts happening. I mean, in type 1 diabetes, for example, some autoimmune, thing that's starting to destroy beta cells gets started. Some T cells start getting overactive and they start, and maybe they bind to other T cells and sort of tell them to become overactive as well. Maybe there's some complicated feedback loop. We don't yet know how that works. But presumably at the beginning, it was presumably some particular thing that seeded that process happening, presumably. I mean, again, it may very well be the case that in many diseases, for example, it's like there are many strikes that have to happen before you're out, so to speak. There are many things that have to all happen before the disease isn't just rolled back by the immune system and other things. I mean, I think the way to think about it, perhaps, is that by the robustness that we have been evolved with in the course of biological evolution, we are imbued with a certain robustness. So many small perturbations just die out in the practicality of a whole organism. I mean, what I've been showing here involves an incredibly simple genome. When we have many different possible cases in the genome, many different subsystems in the organism, it's maybe more likely for one to just roll back through robustness and for a little perturbation to die out. But then what we're talking about is the perturbation that didn't die out, the perturbation that got over the hill, so to speak, and started to roll down to a different attractor. That's kind of what we're dealing with. Let's see. How might we best combine computation and machine learning and the empirical experimental, LC asks, so that they influence each other's research directions and priorities in sort of a virtuous cycle to accelerate discoveries? Well, let's see. I'm going to go on because LC has some other comments. With pockets of reducibility and inherent irreducibilities, are there differences at various levels of emergence and analysis, especially that might be exploitable, like there are in physics? And what about the more systemic cases, both diseases and treatments, where complex interacting perturbations interfere with each other? What are the limits of tractability versus intractability? Yeah, this is an interesting question. What does this mean for what one should do in sort of medical research? And I mean, there are things that we realize are just one thing this perhaps pushes one towards is, personalized medicine is worth doing because it won't be the case that we can see from sort of what happens with genetic variability of the kinds of things we're doing here. It's inevitable that there will be sort of different things happening for at least this sort of idealized model pushes for the fact that there will inevitably be different things happening for different genetically different organisms. And I think that that realization kind of tells one's likely to have to deal with kind of the specifics of particular individuals and so on. I think another thing that sort of pushes one towards is kind of this regenerative medicine idea. It's kind of like the idea that biology uses fundamentally, which is, one generation dies, a new one is born, so to speak. And biology, it's kind of like back in the day, you could go fix your computer, you can open it up and you can change, you can unsolder a component and solder a new component in again. But modern computers are not user fixable in that kind of way. You just pull out a whole module or pull out a whole computer and replace it with another one. And that's kind of the scheme that biology has found as well, of just one generation dies and another one to sort of takes over. Now, the sort of more microscopic version of that is to say, let's take an organ, for example, and replace it with a fresh new organ, for instance. Whether that can be done at sort of a more molecular scale by sort of going in and sort of replacing things, replacing one's mitochondria, replacing some other aspect of cells, extending one's telomeres, whatever else it is, whether one can do that in a way that's kind of across many cells, or whether one is just taking out sort of whole subsystems, whole organs and things and replacing them, I don't think is clear. And no doubt both approaches are relevant. But I think, at some level, I guess this also suggests you kind of have to fight sort of perturbations. The thing that is most likely to work in the end is fighting the perturbations the way the immune system has learned to fight the perturbations, sort of at a molecular level, or at least at an individual cell level. And I think that, and the kind of the large scale thing is ultimately sort of, you can do a certain amount, but it's ultimately not going to be fully effective. I think the other thing that comes through is the idea that sort of the workflow is you take what you observe, the symptoms you observe in this very coarse-grained way, you make a diagnosis that's also very coarse-grained. You say you have disease, ICD-10 code, disease, blah, blah, blah. That's, and then you say, okay, given that code for what disease you have, this is the treatment. This is the treatment code that you need to apply. That kind of sort of very discrete kind of set of possibilities, we can kind of plainly see that's only a very coarse approximation to the best thing one could do. The best things one can do presumably involve more and more data. And certainly my vision for things would be that the idea of diagnosis, you've got this particular diagnosis code in the middle, is something which is a feature only of our times, that eventually it will be more, you go from the symptoms, possibly observed in a very detailed way, possibly observed with kind of, it might even be observed at the molecular level, and very detailed symptoms, then computation for what you do about it, then applying that computational treatment, so to speak, which may be very broad. It's not just take these pills and call in the morning type thing. It's something where it's kind of, here's an algorithm that is going to be run by this collection of artificial immune system cells or some such other thing, or by this molecule that has some kind of algorithmic character to it. Here's this algorithm that's going to be run. Now it goes in and applies itself to lots of cells. And that's kind of what one's going to need to do to kind of fight back this built-in computational irreducibility in these systems. And anyway, a few thoughts about that, but I don't really. I mean, it's a very. There are some suggestions about things that are sort of always, one's always going to be fighting the entropy, so to speak. And there are things where And in terms of, is there a sort of killer direction of computational reducibility that is sort of the great solution for some areas of medicine? Well, we can expect that there will be some of those. We just don't know. It's not clear what they are. And I guess I don't really have a fully formed idea. And I think it starts depending on more details of biomedicine to see how you really attack those things. All right. Let's see. I'm going to wrap up soon here, but yeah, okay. So there's a question from Elsie. What about aging? And what's the story with aging in this kind of picture? I think aging is in some measure can be sort of coarsely parameterized by a series of perturbations. And the question then is, and this is indeed an interesting fundamental question, is aging sort of this computationally irreducible process a bit like the increase of entropy in a thermodynamic system, where it is really you have to fight it with huge amounts of computation to fight it back, or not? I don't know the answer. I think that that's why I think these kind of things that are more at the immune system level, more at the level of every cell of this type, more at the level of replace this whole organ and so on, those seem more promising than the kind of like, drink the elixir of eternal youth and be happy type thing. That it's much more likely that you're going to have to go to, I think the likely takeaway is you need to go to a lot of kind of computational effort to fight back the ravages of aging, so to speak. And it's kind of frustrating. It'd be nice if there was just the sort of the elixir that can be found. Perhaps there are, I think it's going to be a more kind of a more computation intensive thing where the computation really eventually has to be happening at least at the cellular level, if not at the molecular level. Let's see. Ahoy comments. How can this account for the sort of region and global configurations that determine the space of how local rules can play out? E.g., poor mental state can deregulate the whole organism's operation and lead to a breakdown. Yeah, I mean, we see that with these perturbations. I mean, some perturbations kind of eventually have a big effect on the organism and lead to all kinds of sort of cascades of trouble, and others do not. And I think that's the same kind of thing that we often see. There are things where something goes slightly wrong, but the organism doesn't really matter, and other things kind of cascade into big trouble. And let's see. Eric asks, how do you account for fallibilism in our medical knowledge? I'm not completely sure that I know what the word fallibilism means. I think if that means our medical knowledge is sometimes wrong, I think the main takeaway from this is that in medicine, we are fighting computational irreducibility. Computational irreducibility is giving us a torrent of complexity and phenomena and so on. And it is remarkable that we can do anything in medicine. The fact that we sometimes get the wrong answer and so on is, that's not terribly surprising. It's not so simple to come up with medical conclusions. One of the things that's nice, for example, in physical science, we can do these experiments on very kind of controlled, contrived systems. The system that is us is the result of a few billion years of evolution, and its solutions to how to be the way we are involve computational irreducibility. They're not simple solutions. It's not like that simple mechanism where we can just say, hey, the chain fell off your bicycle type thing, put it back on and it'll all be good. We're dealing with a system that has all of these computationally irreducible elements where there isn't that kind of mechanistic solution to how to fix it. Yeah, okay, there's a comment here about definitions of fallibilism. Okay, so that all beliefs are potentially false and open to revision. And the fact that medicine has gone through many oops it wasn't true that ulcers, stomach ulcers were caused by stress. They're really bacterial, those kinds of things, or it isn't, or germs really exist and so on. There are many places where medicine has taken a wrong turn. I think that's really just a reflection of the fact that experiments are hard. Experiments in an area like this are difficult, and there are many sort of confounding factors and so on. It's hard to have kind of the definitive, it's, this kind of simple physics kind of result. It's a very different kind of system that we're dealing with. I mean, I think that the recognition that these things are hard is itself worthwhile. The recognition that we may not know, it may be very difficult to tell, is worthwhile. And while there's a search for sort of very human reasons, there's a search for certainty. We want to believe, oh yes, we know exactly what to do, and that's something which we have to realize. It just isn't. In many cases, there really is going to be. It's going to be very difficult to know that. Let's see. Well, Mornax asks, is it reasonable to assume that the fight against perturbations in a biological organism may eventually be manageable with a computational approach? I mean, I think the main thing to say is you've got to fight computational irreducibility with lots of computation. And I think that that's sort of the main takeaway from this, that just, the thing, and this has been a long story in medicine, that we do very coarse things because that's what we can do, and we kind of say, well, this very coarse thing is just going to solve the problem. That usually it won't be quite the case. There'll be little sort of pieces that are side effects. It may be sufficient. It may sufficiently solve the problem that we feel healthy and all is good, but at some we have to realize that ultimately there will be things that aren't quite that are not quite as we expect. Well, I should probably wrap up here, but thanks for joining me, and I hope I consider what I've talked about here to be just a tiny seed of this kind of computational paradigm and its approach to the foundations of medicine. Medicine has not traditionally had much in the way of kind of formal foundations. This is sort of an attempt that I hadn't imagined something like this would be possible. This was sort of the result of thinking about these models of biological evolution, which I also was kind of surprised were possible. But I think this kind of is I hope, a seed for a lot of what could be developed in terms of a whole sort of pattern of thinking around medicine. I mean, I give the analogy again of Darwinian evolution, for example, where before that time, it's like there are all these different organisms and they just happen to be this way. And after that time, it's like, well, these organisms progressively evolve and we can kind of identify fitness purposes for different kinds of behaviors and so on. And that's, it took a while to develop from sort of the core idea of kind of natural selection and origin of species and so on to kind of the sort of developed view of the intellectual paradigm that is modern evolutionary biology. I think we can actually advance that paradigm a lot through computational ideas, and that's a different thing that I've worked on. But now we have kind of a new domain in which we can kind of have this kind of formal structure, and that's I'm hoping other people will pick this up and think about it, and and maybe build on this kind of paradigmatic structure things which are more directly useful and actionable for practical medicine. And I think that's that's sort of the path forward, so to speak. This is a framework, like other kinds of things, like the framework that one has from, I don't know, mathematics and calculus and so on. It wasn't that framework, after a while, maybe 100 years or so, started to give one kind of engineering solutions for how to do things. But one needed that formal framework to have a place to start from. And that's what I'm sort of hoping this provides, a bit of a seed for a formal framework to start building things in medicine from. So thanks very much. And I think a week from today, I'm trying to catch up on things that I've written. I will be talking about the what I posted about studying the game of life as a model for kind of the arc of innovation and the sort of the meta engineering, the sort of what one can say about the overall kind of structure of the course of engineering. Okay, well, thanks very much, and bye for now.